DNA damages, an overview. When one thinks about DNA damages, the first thing that comes to mind is mutation. By way of definition, mutation basically is when there is an alteration of the sequence of nucleotides in a DNA, and that alteration in the sequence of DNA gives rise to an altered protein from a functional gene, then of course what is understood is that since there is a damage in the DNA, it is being observed at the protein level as well. The question that arises here is, apart from mutations, are there any other form of DNA damages? So this overview looks at different ways by which the DNA can be damaged in a cell. The basic agents, whether they are physical or chemical or biological, are able to carry out damages within the cell to the DNA. Now let us look at what is the learning outcomes of this session. Obviously, the DNA damages that can happen within a cell is going to be understood. The mutations can arise spontaneously every cycle of replication. And what has been observed is that certain sites in the chromosomes are hot spots for mutations. What does this mean? It means that within a chromosome, there are certain regions, there are certain cis elements where the frequency of mutation is very high. DNA damages can remain if during replication errors have escaped proofreading. DNA damages can be due to hydrolysis or deamination, alkylation, oxidation, ionizing radiations, etc. DNA damages can also be due to gene rearrangements. Let us look at the DNA damages that can happen due to errors during replication. It is well understood that whether it is the prokaryotic system or the eukaryotic system, replication in the cell happens because of the DNA polymerases. And every DNA polymerase has an inherent 3' 5' exonuclease activity that allows it to proofread whatever it has synthesized. In other words, DNA polymerase 3 or DNA polymerase 1, or for that matter, any DNA polymerase has the ability to proofread and correct whatever wrong nucleotide it has added. And despite that, errors are possible. Let us consider a short oligonucleotide which undergoes the first round of replication. Now, as it undergoes first round of replication, obviously this is going to be one template and this is going to be the second template. And using these two templates, the DNA polymerase, say for example, is synthesizing the new strand. So this strand was the parent strand, and on this strand the new strand has been formed, but as it synthesized the DNA, instead of adding the correct nucleotide, it has added a wrong nucleotide over here, leading to what is called as a mismatch. Therefore, although the DNA stays as a double-stranded DNA, at this portion where the base pair is not complementary, you will see that the DNA bulges out a little. Therefore, there is a mistake that is there over here due to the DNA polymerase 3 not having proofread this mistake. In the other case, using the other template, the DNA polymerase has not made any mistake, and therefore the new daughter DNA that is formed is a correct daughter DNA. If this is the case, everything is going to be normal. However, if this is the case, two things may happen. One is that this DNA, because there is a mistake over here, can be recognized by a repair mechanism and can be repaired. Or what can happen is that it escapes repair, it has already escaped proofreading, and after escaping repair, this DNA, when it undergoes a second round of replication, one of the strand will have the original template base, while the second strand will have the wrong template base. So if that is the case, the strand as a template which had the correct template base. And if the DNA polymerase does not make a mistake again, then in all possibilities, the second daughter DNA that is formed is going to be absolutely correct. And this base pairing is the same as this base pairing, which means that after the second round of replication, this DNA is normal. And so the cell that acquires this is going to be normal. While in the other case, where you have already the template having G, which is not present in the original over here. So this G has come because of this wrong nucleotide having been added. And so when this template is being used to synthesize the daughter DNA, what will be added by the DNA pol is of course the complementary to G. But the end result of this is that instead of having AT base pair, one gets what is called as a GC base pair. And so this becomes what is called as a point mutation, one base pair having been altered. And any cell that has this altered nucleotide base pair is considered to be a mutated DNA. Therefore, if this mutation were to happen in the germ cells, it would be inherited. But if this mutation were to be remaining in the somatic cells, then it would be present in that generation, but it may not go to the next generation. Now let us look at DNA damage that can happen because of spontaneous hydrolysis. Before we go into understanding how or what damage is done because of spontaneous hydrolysis, hydrolysis happens because of the presence of water around the DNA. And therefore, it is very ironical because the DNA achieves or gets to its stable conformation because it is present in the aqueous environment. So, water is a natural surrounding of the DNA. And yet, it is this water that is able to carry out hydrolysis and remove certain functional groups from uh, say the phosphate, or say for example from the sugar, or even from the nitrogen bases. So from the nucleotides it is possible that a functional group is hydrolyzed by the water. Now let us consider citidine. So citidine basically has a cytosine with an amino group present over here. What is observed is, in presence of water, the water can spontaneously remove the NH2 by hydrolyzing this bond, and what remains is a O over here because of the water causing the hydrolysis. Now when you look at this nitrogen base is now uracil. So what has effectively happened is that cytosine has been converted into uracil, and so the nucleotide that you're getting is uridine. Now, uridine is an unusual nucleotide in DNA. What is usual in DNA is when the DNA has thymidine. And in fact, probably that is the reason why the DNA has adopted to have thymidine in its structure rather than uridine. Because a naturally present citidine, if due to deamination gets converted to uridine, then this uridine in the DNA will be considered to be unusual. Why is this important? It is important because only then will the DNA repair system recognize it as something not normal, and therefore they may need to correct it. So therefore the presence of thymidine, okay, which is normal in the DNA, and the presence of uridine, which is unusual in the DNA, is basically accounted for. So we consider over here therefore, where originally you had a GC base pair, correct? The C having got deaminated to convert it to U, after the first round of replication, what you will get is AU rather than GC. So because the C has got converted to U, in the new daughter strand, you will not get G, but you will get A. So you can see there is an alteration. There is a mutation that is happening because of spontaneous hydrolysis. of what? Of citidine, that gives rise to what? Uridine. Now let's look at another similar example with slight difference. Many citidines in the DNA can exist as methylated citidines, and the methylated citidines are, say for example over here, you have the 5-methyl cytosine. This looks very close to being thymine, except for the fact that you have an amino group present over here, and therefore this is 5-methyl citidine. Now, if the water were to hydrolyze this bond, and on hydrolyzing the bond, the ammonia is the ammonium NH2 group, amine group is released. It leaves a double bond O over here. So therefore, what does this become? This becomes thymidine. Now, thymidine is a usual nucleotide in the DNA, and hence it may not be recognized as abnormal by any repair system, and therefore it remains. But originally, what was it? It was a citidine that was just methylated at the 5th carbon. So this kind of mutation is not really good for the system. Why? Because it cannot be recognized as an altered nucleotide, and hence it can remain without being repaired. Such 5-methyl cytidines are considered to be hotspots in DNA. So basically trying to say that these are the regions, or these are the nucleotides which can undergo hydrolysis spontaneously very frequently, giving rise to the change of cytidine into thymidine, which means that after first round of replication, where T is the altered one, initially it must have been 5-methyl cytosine, now it has become T. So therefore the new daughter strand that is going to be formed will have A. So instead of GC base pairing, you would have an AT base pairing, and this is considered to be transition. Okay. So therefore what is clearly observed is that just simple hydrolysis of the bond that holds the amine to release the amine and replace it with double bond O is going to lead to deamination, which in turn is going to lead to an unusual or a changed or an altered nucleotide in the DNA. This is therefore one way of DNA damage. Now let us go to another spontaneous hydrolysis that is observed. Say for example, you have a guanine. That means you have a guanine nucleotide. So in this case, what is observed is, suppose spontaneous hydrolysis happens and it is cleaving the N-glycosidic linkage, thereby the guanine is released, and what remains is the sugar phosphate alone. So which means that a base from this position has been totally removed. Such a process is what is called as depurination, and this leads to a place where there is no nucleotide at all. You see that the purine has been removed. So at this point, you have no nucleotide present. So therefore, this is called as an abasic site or apurinic site. Now, this is also therefore unusual in the DNA, which can be recognized by the DNA repair system and repaired. Now let us go to another type of DNA damage, which is alkylation. And what has been observed is guanine is a common target of alkylation. So if this is the guanine present, then alkylation is possible at this end, at this end and at this end. So this is basically the 3rd end, the 7th end and the 9th end. And you also have this part of the guanine that can undergo alkylation. Let us consider that the alkylation has happened at this point, and what you're getting is OCH3. So this is alkylated. So what you get is O6 methyl guanine. Okay. Now interestingly, a guanine which binds to cytosine, but when it becomes O6-methyl guanine, instead of complementarily binding to cytosine, it binds to thymine. And therefore, a G and a T is an not normal base pair, and therefore it is an alteration in the DNA sequence. So what you have is a GC base pair becomes AT after replication, and therefore that is a change, that is a point mutation. You can also say, for example, I have the DNA exposed to ionizing radiations and several different chemical agents that give rise to free radicals. And these free radicals are able to affect, say, for example, guanine in such a way that at this point, okay, you have the 7 and the 8th position where you have a 7,8 dihydro 8-oxoguanine formed, which is termed as oxo-G. Now, therefore, what was a double bond over here is no longer a double bond because of these free radicals. So, the free radicals have added an electron over here, and because of that highly reactive electron, the double bond is broken, and in place of that, 2 hydrogens are placed. Therefore, 7,8 dihydro 8-oxoguanine. This is commonly termed as oxo-G. Now, this alteration, just because of the free radicals, oxo-G can bind to cytosine, but instead of 3 hydrogen bonds, it binds to the cytosine with 2 hydrogen bonds. And plus, oxo-G also has the ability to bind to adenine. So therefore, this interaction is definitely going to be an unusual interaction, and therefore you can already see an alteration present because of the formation of oxo-G. Therefore, in this case also, a GC base pair will ultimately after replication become an AT base pair, which is definitely a single point mutation. Therefore, what is also observed is such mutations have been commonly found in many human cancers. A guanine can also be deaminated, and so here you will have an O present after the NH2 is removed. So therefore, this also becomes an altered nucleotide. Now let us go to another type of DNA damage, and that is something that is called as pyrimidine dimers, and this happens when the DNA is exposed to UV light. What is observed is that DNA is able to absorb light at 260 nanometers. 260 nanometer falls under the UV range of light. Why does the nucleotide absorb light at 260, or why does the DNA absorb light at 260 nanometer? It is because the nitrogen bases of the nucleotides in the DNA have ring structure. And because it has a ring structure, it has delocalized electrons. Those delocalized electrons can absorb light, and they absorb light of energy such as equivalent to 260 nanometers of light. Therefore, they absorb that light, and on absorbing the light, sometimes what has been observed is that if there are two adjacent pyrimidines present, especially if there is a thymine and a thymine present, then between the thymine and the other thymine, you have a cyclobutane ring formed. So the fifth carbon and the fourth carbon of one of the thymine is able to bind to the fifth carbon and the fourth carbon of the second thymine. Thereby you have what is called as a cyclobutane ring formed between two thymines. So now it is no longer two separate thymines, it becomes what is called as a thymidine dimer. The question that comes up over here is that why is this TT, or if it is a C present over here and a T present over here, you can also have a CT adduct. Now the question that is going to be very pertinent here is how does it affect, or what is it that leads to a DNA damage because of a thymidine dimer formation or because of pyrimidine dimer formation? So the answer to that is when you have thymidine dimers present, it has been observed that it is an impediment to replication. It is also an impediment to transcription. What has been generally observed is that when the DNA polymerase is synthesizing the new daughter DNA, say for example, it has recognized A and it will introduce a T to complementarily bind to A. When it comes to a thymidine dimer, DNA polymerase 3 or polymerase 1 is not able to understand what it has to add because this structure or this conformation does not conform to any of the nucleotides. It does not conform to thymine alone, it does not conform to uracil alone, it does not conform to guanine or a cytosine alone. So therefore it does not know what to do with this thymidine dimer. What should it add? So it may randomly add something and move ahead, or the replication fork will just simply arrest at this point. And therefore, unless this has corrected, the replication fork may not move ahead. If this is not corrected, then the replication of the DNA is going to stop. If the replication of the DNA is going to stop, cell division will not happen, right? So it is going to lead to cell death if it is not being corrected. So therefore it has been observed that if a cell is exposed to UV for a long period of time, then the cell may undergo cell death. And therefore the very simple logic of why UV light is used to sterilize many areas. For example, when one wants to carry out sterile work, then the UV lights are put on in the room or put on in the area where the work is to be done for at least 45 minutes before the work. So that is just basically to ensure that if there are microbes that are present on the surface of anything that is going to be used in work, the UV light is able to ensure that those cells are dying. So therefore, exposure to UV light is considered to be damaging to the DNA. Till what extent you expose the DNA to UV light does matter. Okay. Now, you have another observation. There are many chemicals, reactive oxygen species, ionizing radiation, nucleases that can lead to what is called as breaks. So you can see this is a double-stranded DNA, and a double-stranded DNA, because of any of these, has what is called as a complete break across the two strands. So this is what is called as a double-strand break. Double-strand breaks are very detrimental to the cell. If they are not repaired, the cell is definitely going to die. So what is this? This is not a mutation. This is, however, a DNA damage. When the DNA is broken completely into two, then it is a damage. When you have only one strand of the double-stranded DNA broken, that creates a nick, and that nick also is going to be considered as a DNA damage. If there are repair systems that can take care of this, they are going to take care of this as well. Then we come to a chemical agent that can be used to induce or that is responsible for mutations in DNA. Base analogues. What are base analogues? Base analogues are basically molecules which have a very similar structure to one of the nucleotides or one of the bases. So say for example, you consider 5-bromouracil. The 5-bromouracil is very similar to thymine. So if in the template strand there is A and the media or the media in which the cells are growing has 5-bromouracil, the cells will take up 5-bromouracil and the DNA polymerase can mistake the 5-bromouracil for thymine, right? So instead of thymine, it will add 5-bromouracil in place of thymine and think that it has done a correct job and move ahead. But what is interesting is that the 5-bromouracil, which is an analog of thymine, does not bind to adenine, but it binds to guanine. And since it binds to guanine, effectively it is going to be a wrong base pairing. Because you cannot have T bound to G. You will have T bound to A. So therefore, after replication, instead of an AT base pair, what you will get is a GC base pair in the second round of replication. There is another base analog, that is 2-aminopurine. The 2-aminopurine is able to, when introduced into the DNA because of the uh, DNA polymerase, considering this as adenine, okay, it adds it, and so this 2-aminopurine is able to bind to thymine, which is okay. But when the 2-aminopurine binds to cytosine, then this is again a mismatch. So it will lead to, in the second round of replication, a mutation. So this is how base analogues can actually be used to induce mutation. That is because the base analog competes with its base to be taken up by the DNA polymerase to add to the newly synthesizing DNA. Then there is another chemical group of chemical agents which are called as intercalating agents, and that also can cause DNA damage. So you have what is called as ethidium bromide, you have proflavin, and you have acrid in orange. These are considered to be common intercalating agents. Now, what is the end result of these intercalating agents interacting with DNA? So what is observed is that they can cause deletions or insertions, and as one knows, deletions and or insertions are mutations eventually. So what is observed is that many intercalating agents can stack itself between two base pairs. So these are the original base pairs in the DNA. Now within or between two base pairs, they have the either the acrid in orange or the ethidium bromide can stack. So you can see this picture is basically showing different types of drugs that can actually intercalate within the DNA. Now, why or how does this cause deletion or insertion? So suppose you have an intercalated agent present in the DNA, and if this portion of the DNA unwinds to two strands, then you still have this intercalated agent present. So when the DNA pol is synthesizing the new DNA and it comes to the intercalating agent, it does not know what nucleotide to add for this intercalating agent. So there are two possibilities. One is that it is possible that this intercalating agent is shielding this nucleotide. So as a whole, this can be such that the DNA pol does not recognize a nucleotide and therefore does not add a nucleotide here and moves forward with this. So what happens is one nucleotide over here is not added, and therefore that can lead to deletion. Or what can happen is that this intercalating agent can be considered as a nucleotide. Against this, a nucleotide is added over here, and so you have now 1, 2 and 3. So where you have 2 originally, you have an extra nucleotide present in the new daughter DNA, thereby leading to what is called as insertion. Interestingly, because intercalating agents are responsible for deletions or insertions. Say, for example, a, a cell which has become cancerous and has lost the regulation to stop dividing. So it is continuously dividing basically. Many of the intercalating agents have been used as drugs to kill the cancer cells because introduction of deletions or insertions can lead to damages, a lot of damages in the DNA, and if the damages are not repaired, then the cells will tend to undergo cell death. So therefore that becomes one way of treating the cancer cells. So this is something that is being taken as a basis of treatment. Another interesting fact is that when intercalating agents get intercalated within the DNA, okay, you will find you have one agent here. Suppose you have one more over here and one more over here. What does indicate that the number of base pairs per turn will not remain the same if you have these intercalating agents. So what does it mean? It means that it is affecting the twisting number of the DNA. And if it is affecting the twisting number of the DNA, it is affecting the DNA topology, right? So the question that arises over here is whether it is introducing a negative supercoil or it is introducing a positive supercoil. But it is definitely causing change in the twisting number, which in turn is causing a change in the supercoiling. Please keep another thing in mind. Ethidium bromide is a very commonly used staining dye for DNA. Since you have the ethidium bromide getting intercalated in the DNA, wherever you have DNA, you would have the ethidium bromide present. Now, ethidium bromide under UV fluoresces. So wherever you will have DNA present, there you will have ethidium bromide. And when you shine UV light on it, that part, because ethidium bromide is present, will fluoresce. So wherever ethidium bromide is present, there the DNA is present. So in effect, you are staining the DNA with ethidium bromide. So it is a very commonly used agent in DNA studies. But like mentioned, because it can cause deletion and insertion, it is carcinogenic. And therefore, while handling ethidium bromide, one has to be extremely careful. One ought to wear gloves while handling ethidium bromide. One ought to discard ethidium bromide very carefully to ensure that nobody is exposed to ethidium bromide. So safety measures have to be taken while working with these intercalating agents. One other way of DNA damages is gene rearrangements. Now, there are various ways by which gene rearrangements are possible. For example, homologous recombination, okay, is a way by which you can have rearrangements. But just to observe, suppose you have a part of chromosome A and a part of chromosome B getting dislocated or translocated. So this comes here and this comes here. So there is a gene rearrangement, as you can see. This is may not be good for the cell, may be good for the cell, one doesn't know, but this is a rearrangement and therefore this is considered to be a DNA damage. Okay, simply deletion can also lead to a DNA damage. So for example, all genes that were present in this have been removed because of breaks in the chromosome. The breaks in the chromosome can be because of ionizing radiations, and so this deleted genetic material makes the chromosome shorter, and because the chromosome is shorter, there are several functionalities that have been lost because the genes present in this have been lost. Okay, so gene rearrangements are at the chromosome level. There are different kinds: deletion, duplication, insertion, inversion, translocation, etc. So here, just to understand what is gene rearrangement, suppose you had in a DNA gene A followed by gene B and followed by gene C. If there is a rearrangement, you can have BAC. So this is considered to be a gene rearrangement, and this is something that can happen in DNA. So let us conclude. An error of adding a wrong nucleotide, and if not corrected, then it leads to a mismatch that can be seen as a mutation in the second round of replication. Spontaneous hydrolysis can lead to numerous DNA damages like deamination, depurination, etc. Ionizing radiations, chemicals, free radicals can cause oxidation as well as DNA breaks. Base analogues, intercalating agents, UV irradiations can all lead to DNA damages that can be manifested into diseases or impairments, and at the same time can be used to treat diseases like cancer, where stopping DNA from multiplying helps in decrease in proliferation. Thus, DNA damages are a natural phenomenon within cells which may be repaired or may become permanent, leading either to detrimental effects or evolutionary effects or no effects at all. Thank you.